We've set the scene. There's lots of new medicines in development. How are we going to ensure they're safe? So the next talk is by Victoria Hutter. Victoria is the Chief Scientific Officer at ImmuONE. It's a contract research organisation specialising in vitro toxicology solutions for the inhaled products industries. She's also Professor of in vitro toxicology at the University of Hertfordshire. And Victoria and her team developed new models and assessment methodologies for inhaled toxicology with a focus on the inflammatory responses of the airways and the alveoli.
Cool. Thank you very much, Ben. Thanks for the introduction, and thank you for inviting me to talk today. So I'm going to give you a whistle-top tour of in vitro inhalation toxicity. So kind of all the key aspects that we need to consider and the context in which it can be used and how we can kind of change the landscape of in vitro toxicology. So the four main things that we need to consider: the model, the type of model. Is it the right model for the application? There are lots of different types of models on the market, ranging in complexity, and we will cover those. The exposure. So there's lots of different ways we can expose in vitro. Some are more physiologically relevant than others. There's a whole plethora of different assessment methodologies, but picking the right one with the right endpoints and the right interpretation is also really key. So we'll go through these four areas in the next 20 minutes. Carsten really nicely summarised what the kind of the physiology of the lung, and just to really highlight that understanding where deposition happens in the lung and choosing the right model in vitro for your toxicity assessment is really key. You've got very different specialised functions right from the nose down to the alveolus, and depending where that deposition happens, obviously you need to pick the right area of the lung. And it might be that you have to consider more than one model of the lung. And the bronchial barrier is quite different to the alveolar barrier, and the leakiness of those membranes is quite different. The physiology and the different types of biological barriers that the cells, that the particles might interact with is very different. So selecting the right model is really key. And not just the cells of the barrier. A lot of people overlook the lining fluid. This can be quite an important barrier, particularly for things that are quite biologically active or quite insoluble. The proteins within the lining fluid can have an influence on toxicity, or at least the presentation of that molecule to the cell to determine the final response. So in terms of models, we can go through from quite straightforward, simple 2D epithelial cell culture models, which have been around for now 20, 30 years, quite standard assessment. We can go through to increasing complexity. So over the last 20 years, it's kind of been well established if you grow cells at the air-liquid interface, you can differentiate them into something that looks more physiologically representative of the lung. We can move to primary cells. We can have primary cells that are kind of more commercially available or just single cell lines, single strains. Moving up to cold culture, more complex 3D systems like spheroids, and then finally lung on a chip. So I'm going to go through these in a little bit more detail, kind of advantages, disadvantages of all. There's no ideal model, and normally it's like thinking about the question that's being asked and picking the right model for answer the right question. So cell lines, probably the easiest, most well-established way. These are normally, they're continuous cell lines. They will grow and divide regularly. They have a kind of infinite lifespan, and they're normally acquired through cancerous sources or through induction of viruses and genes that prolong proliferation. Whilst they can be homogeneous or heterogeneous, there's a diversity of responses within that population of cells, which is important to consider. And there's lots of different lung-specific models. Previously, we used to use CACO2, so an intestinal model to mimic the lung, but there's definitely been a move to using more lung-relevant cell lines. And there's a whole raft of the ones that can be used. Just because they're cancerous, obviously they're a really good model of the cancerous lung cancer, but as long as the functionality that is there that you're looking for, so if you're trying to assess if something is alive or dead, or if there's a particular protein, then they've still got a good use. Moving forward to kind of more complex models, so growing cells at the air-liquid interface. About 20 years ago now, it's kind of well established that if we can grow cells on hanging transwell baskets and expose them to an air-liquid interface at the top, then they will grow and differentiate. They will become, I'm not going to attempt to use that, they'll become more ciliated. They'll have a taller epithelium, more columnar epithelium. They'll have a change in their secretions, and their barrier function will also change to look more physiologically representative. The primary cells come from donors. So these can come from a single donor, so they can be bought from a company. A single donor, you can find out the age of the patient. You can have disease models, maybe ones that are COPD related. These termed as more biologically relevant. Their physiology should be more similar. The proteins they actually express should be more similar to an in vivo patient. But they do have a short lifespan, and they are quite variable between patients. So using as an initial screen to understand toxicity or mechanism might not be quite so appropriate. There's also commercially available tissues, and these tend to combine a pool of donors. So you've reduced that variability between patients, and they can also combine a number of different cells. So they can be the upper airways, the lower airways, or combined with fibroblasts or different tissues within the lung. Yes, the next complexity is then to build on that, so have multiple types of cells present in a system. So we're not just looking at epithelial barrier and the potential of that to break down, but also looking at the endothelium, the fibroblast, the immune components, so macrophages, dendritic cells, and how cells will interact with each other, which gives a much more kind of a tissue complex environment just as a single cell. So the cells should be selected depending on the response mechanism. So there are some nice alveolar models that are out there with companies like Matek or co-cultures of primary cells. There's also, we can do it with cell lines. So there's lots of in the literature. There are some that are on the market that use different types of epithelial cells with immune cells. Some of these are in a single system. Some of these can be pulled apart. But the co-culture model is really to understand kind of more mechanistic behavior and how those cells interact with each other. Moving on, there are spheroids and organoids. So these will be self-assembling cells, normally from stem cells or progenitor cells, and they self-assemble either when giving a scaffold or without the need of a scaffold. These are much more in the literature. So in terms of having a standardized platform for understanding toxicology, I would say they're definitely in their infancy. But it's something to look forward to the future because it's an isolated ball of cells that acts like a tissue in a discrete entity. And then I guess the kind of moving towards the gold standard is having an organ on a chip. So something that it will simulate human physiology within the system. So in the lung, mechanical forces are really important. So you're constantly breathing, your cells are under different stresses, and also the physiological flow of either air or blood. And the idea about this is the similar principle to the air-liquid interface with cultures, that they produce a closer human physiological representation of the cells or tissues. So just as what is physiological relevance? So particularly for the alveolus, we've got the right cells, that we've got the right exposure conditions, that the blood flow is mimicked, so the constant flow of blood can affect the physiological differentiation of the cells, that we've got kind of a right surface area to volume of cells, the ratio of the cells in the tissue is correct, and that we've got a stretching mode, so that stretching kind of stress and strain is appropriate to the physiology of the lung, and that can change how the cells respond and how they differentiate. So there's some examples of organ on a chip. So this is one by Emulate. The cells are grown in a channel. So the cells are grown on the blue surface, which is then exposed to an air-liquid interface. And there's a channel of the air can be dragged across the top. And then there's a channel at the bottom, which will represent the blood. And that's coated with endothelial cells. Immune cells can be put into this as well. And the basal media will flow through that system at a physiologically relevant speed. There's also flex within that membrane. So that membrane can move as you want it to represent the lung. The second option is another one. It's one that's on the market, and this has a very stretchable membrane. So it's really for mimicking breathing. So it goes within a system that has cyclable breathing, so about 12 cycles per minute, about 8% stretch. And these have been shown to be used with cell lines and with primary cells to have a more physiologically representative model. So moving away to kind of in ex vivo models, precision lung slices are something that have been coined particularly by IIVS in the US. So these are long slices which are cut. The organ is collected, it's filled with agarose, and then it's sliced in very, very thin slices, frozen, and then can be shipped for use. And they contain all the different types of lung cells present, so it gives that real idea of the 3D environment of the cells at the time of slicing. And they can be maintained for a number of weeks. And can be used to look at acute, or I said chronic, but slightly longer term exposure. I guess one thing that I didn't mention before with the other in vitro models is the time of culture. So some cell lines can be used for a number of days. A lot of the kind of co-culture models can be used up to a number of weeks, and some of the primary models can be used for a number of months or even up to a year. So we can look at both acute effects, but also kind of chronic or longer term repeated dosing as well. So how do we pick the model? It's getting the right types of cells for the right response is really important. Not every cell will be optimally optimized for every different assay that you do. There's a lot of evidence that some models will be very good at responding to an inflammatory response. Others will give you a much more idea of a certain protein is present. So picking the right cell for the question is very important. Making sure it's compatible with the right techniques in terms of understanding how that cell is going to be assessed at the end is quite key. Making sure that it can be exposed in the right way. Not every system, for a 2D cell system is much more difficult to expose to an aerosol than something grown on a transwell. Again, an organ on a chip needs to have the right way to expose it in the way that you need to. It needs to be compatible with the assessment endpoints. So if the model needs to be separated, or if it needs to be embedded within a system that can assess it, that's also key. And increasing complexity isn't always needed. If there is a basic question and it's a tox-related dose escalation that can be done very simply in vitro with a concentration curve, there's no point of doing that in a very high expensive model, or starting off with inhalation dosing. So thinking about the question that's being asked and picking the right model to answer that question is really key. Just because you have 10 different cell lines present doesn't mean it's a better interpretation. And also consideration of the skill of the end user. Some of these systems are very expensive. They take a lot of time to optimize within a laboratory. So making sure that you've got the right piece of equipment at the right time. So moving on to exposure. So the human lung physiology is quite complex, as Carsten alluded to. A lot of the in vitro tox we do in animals is in rat. Rat and human lungs are very, very different. The easiest way is obviously to put a cell culture media on the cells, but that isn't really representative of what happens in vivo. Ideally, it'd be better to deposit the particle as an aerosol. And understanding that interaction with the lining fluid is often kind of overlooked. And getting the right accelerator exposure can be quite tricky. So just in terms of exposure, if you're thinking about a particle, depending on where that particle lands, depends on how it's delivered. So inertial compaction happens obviously in the upper airways, in the nose, in the trachea, in the back of the mouth. But when we're depositing particles in the lower lung, they form like gravitational sedimentation or even diffusion right in the airways. And getting that exposure and mimicking that exposure can be quite challenging. It's probably only in the last 5-6 years that the equipment has advanced enough for us to be able to do that reliably. And thinking about deposition, when you inhale something, it isn't distributed evenly throughout the lung at all. So this is just an example of an inhaler and some modelling. So this is the throat and the back of the throat and then the top of the trachea. And the way the red spots are the high areas of concentration of where the molecules landed. So when we're thinking about how we dose, we have to dose for the worst case scenario. What is going to be the highest concentration that's going to be deposited? So it's not as easy as taking the dose in the lung, taking the dose that is in the inhaler per puff, dissolving that in the volume of fluid. There's a much larger dose range, and that dose range will vary depending on the patient and the exposure, how they use their inhaler. So dosing in vitro is taking a whole lung system and dosing it on a small piece of plastic with some cells growing isn't always straightforward. There's some deposition modelling techniques and mathematical algorithms we can apply. So we can look at the total surface area of the lung, we can look at the lining fluid, we can do an average distribution. Or we can look at what's more likely to actually land in certain regions of the lung, looking at kind of percentage of deposition within a certain area. But we always look at a broad range. So we always push the system towards what a toxic effect is and scale back to where a non-toxic effect is, and to use that as a kind of evidence approach to say, actually, this is the worst case scenario. So if dosing by solution is quite straightforward, but dosing by particle or aerosol is much more difficult. So rather than having a concentration, generally it's a concentration per area, which mimics the area of the lung and the in vitro area within the transvar. So Carsten said before, but in the alveolar space, it's about 100 meters squared, whereas in the in vitro area, we're talking about a third, or 0.33 centimeters. So getting the right dose, small enough quantities, or correct way is quite challenging. So as a solution, a suspension, generally quite straightforward. As an aerosol, a little bit more challenging. But there are very nice bits of engineering that are being done to kind of help us expose systems in a more relevant way. So dosing in solution is quite certain. As long as it's pretty aqueous soluble, there's not too much non-specific binding to the plastic, that it's not metabolized immediately by the cells, or there's no interaction with the cell culture medium. Generally, what you put on the cells is what the cells are exposed to. But it is less physiologically relevant. You don't normally dose a patient by shoving lots of liquid down their lungs. It wouldn't be deemed ethical. So you're kind of getting effects of toxicity that you might not see otherwise. Dosing using aerosols is much more physiologically relevant. Liquid aerosols are probably the easiest way to dose. And there are several systems out there that can dose between kind of 15 to 300 microliters of nebulized volume, and that exposure can be relatively short, around 5 minutes. So it's the solution's put in a chamber, that's emitted as a cloud, it's homogeneously mixed, and it's allowed to settle by gravitational sedimentation on the cells, which is great. The only problem with some systems is that it's not direct to the cell, so it's in a chamber with several cells exposed, but there are balances in there which can tell you the deposition of the cell. But still, there is variability within that system. So if you're trying to deliver a very, very accurate dose to a cell, it's not going to be as achievable, or it's going to be much more challenging than it is in a solution-based dosing system. There's lots of other things. This is a Cultex, so this is where the dry powder is used, and it's directly put on the cells itself. So reduced variability, but the throughput's much lower, and these systems can be, again, quite expensive. There's another system where dry powders are placed in a loading system, the expansion chamber is sealed, that aerosol is generated, and the powder lands on the sample. So they are possible, but using them in the right context of in vitro tox study is also quite important. You wouldn't be using it in early phase discovery where you're screening lots of compounds, but when you're looking at formulation toxicity or formulation response, definitely they're worth context there. And dosing continuously, this is a drug delivery conference, but there's also options to look at kind of occupational exposure to things like gases, complex mixtures over a number of hours in terms of grams of material rather than milligrams that we're not used to working with. So there's lots of different ways of dosing cells, and the technology for those is really, really improving. So moving on to the assessment. How do we assess? The normal question is, does it kill cells? Is it so toxic that we don't want to be breathing this in? And if the answer is yes, it's probably a no-go. If it doesn't kill cells, great, but that doesn't necessarily mean it's safe. How does it affect tissue cell behavior? Does the function change? There's lots of different functions we can look at, but generally, if that's okay, then no, that's great. It doesn't mean it's completely safe, but it means we've moved a bit further on. If it does change cell function behavior, but it's not necessarily adverse, perhaps it's adaptive, can the cells recover? No, then okay, the weight of evidence isn't great. But if they do recover, excellent, but we need to show that evidence. So in vitro assessment can be used for screening, but it can also be used for mechanistic assessment. So rat studies and vivo studies are great for kind of a whole system and animal base, but understanding the mechanism of toxicity or the mechanism of reaction is quite difficult. But cell-based models are really, really beneficial in this. So the kind of questions I'd ask as a toxicologist is, well, how does it kill cells? How is function affected? Do they recover? And that's linked to something we call adverse outcome pathways. So these are biological pathways of individual events within a cell that would happen that would link to a toxic event. And this is where in vitro assessment can really come into its own to answer those questions. There are standard assessments of epithelial toxicity. So does it kill cells? So basic viability cells that are on plates, looking at the barrier, so using electrical resistance, but also looking at histology. So taking slices of those tissues and understanding, well, how does it look? Does it look like the barrier is affected or the cells are dead? There's several other assessments, so more kind of mechanistic. So Epithelex is a company that have done a lot of work looking at cilia beating frequency. So how fast do those cilia beat? And we can put beads on there to see actually how far are they clear? Do those beads move? Can we link it to things like mucociliary clearance when we add a drug? How does it link into those kind of functional toxicity assessments? Things that are definitely moving forward are not just cell death, but actually what's the underlying inflammation? Is there other cell stress that's there? And there's a range of different things that can be looked at, so cytokines, surface markers, growth factors, so very much antibody-based techniques. And these can be done as an average of the whole cells in the population. But what's of real value is when you're looking at mechanistic information, that each cell responds differently, and understanding how each individual cell responds is quite key to kind of that early mechanistic piece. So if you're looking at average population data, if only 10% of the cells are acting in a certain way, that's masked. But using things like flow cytometry or high content image analysis are really powerful because you can then look into subsections of subpopulations of cells and understand the mechanism that's going on. So it's very much I'm very much in the space of mechanistic-driven assessment. So what is the mechanism of death? And what is the mechanism of inflammation? And we use a whole wide range of techniques. There's genomics, transcriptomics, proteomics, is probably where I work the most to understand what is happening at the cellular level. And the approach that I take and the way the field's moving is definitely towards this multi-endpoint analysis to understand mechanism-driven risk assessment. So not just getting one endpoint out of an assay, but getting multiple endpoints out of assays, understanding how different cells within a system respond. So this is an example where we use macrophage cells with epithelial cells. So from the macrophage, you can really understand what's the toxicity, what's its effect on the metabolism, what's the morphological changes within that cell, what are the biochemical changes within that cell. And we can copy that alongside what the barrier changes, what's the viability, what's the morphology, what's the interaction between the two cells, and we can expose it in different ways to understand that. So multi-endpoint analysis for risk assessment is really key. I was going to give a very short overview of kind of an area that we've moved into in the last couple of years is cell painting or high content imaging. And this is a morphological approach to understand really detailed visions of cells that can be correlated directly to in vivo. So one of the difficulties between the relevance of in vitro assays is that the endpoints aren't often in vivo animals. But with this technique, you can stain cells, you can compare it with histology, and really drive it towards that mechanistic risk assessment. So something that we do quite a lot, or I've been doing a lot, is understanding that histology of a cell. So the image on the right, where cells are highly vacuolated, to understand that approach. But it's not the whole cells in the population will have that response. So using a phenotyping fingerprint, so we can take several different characterizations of the cell, overlay each other to understand actually how does that cell population change individually. The control is quite standard. There's quite a shift in the known toxicant. We can then benchmark this against test items to understand their toxicity. There's just an example of an air control and a known toxicant. And when you're looking at average data, you can't see any difference. But when you look down into the individual cells, you can see here that the phenotype of these cells was much lower metabolic activity and combined with an increased cell area, which kind of indicated an autophagic response, which was actually the mechanism of action of the cell death. And another example where you might see a response that's increased activity in cells and increased stress. So if you're going to add a trigger to a cell, it's going to make it stressed, even if it's just a bit of PBS. But actually on its own, that's fine. But when it's in context with other kind of phenotypes that have changed the cell size and the cell area, that would indicate a more toxic response. So interpretation and context is really important. The purpose of the in vitro test and how we're applying it to safety decisions in vitro is really key. So do we want to differentiate response? Is it that you're looking at test substance A versus test substance B to understand it's something different? We can benchmark response, so we can have a set of compounds that we know are safe, and we can benchmark or draw a line saying when anything below those figures indicates a safety issue. Or we can start correlating response directly to other assays, so in vitro assays to in vivo assays. IVIVC is something that's always a big question. Well, how do we correlate? We need to pick the right markers, the right endpoints for those assays, and to have an appropriate compound set with different in vitro that's well characterized in vivo to be able to build that data set. We're definitely moving towards mechanistic-driven risk assessment, so I talked about adverse outcome pathways and understanding that process. And finally, there's now integrated approaches to testing and assessment, or AATA, and there's some really nice case studies, particularly within the inhalation space of pesticides, so chlorothalonil, where in silico and in vitro and in vivo tools were used to understand, really refine that risk assessment. And finally, one other thing to consider is that we often go from rat in vivo to human in vitro, and making that jump is quite a large jump to make. But using a combination of different techniques, different models to take us through that journey to understand if there's a species difference or a model difference or an exposure difference that's caused that toxicity is quite key. So in summary, giving you a bit of a high-level overview of all the different types of in vitro models, the dosing systems, the considerations that we have to make. Defining that question and the purpose of the in vitro assessment is really key. And there is definitely potential for in vitro respiratory tox and understanding how this can replace in vitro models in the future. I don't think we're quite there yet with completely replacing animal testing, but absolutely reducing the number of animals that have to go through inhalation toxicity testing is well underway, and in the future, hopefully, we'll have to use more relevant models. And that's the end of my talk.